Hey, we're going to talk about Dihybrid cross and law of independent assortment. So, as I alluded to last time, homologous chromosomes get separated by meiosis, right? So you have one chromosome here, one chromosome here. They carry the same genes, so they have the same uh, location. If you have eye color here, you have eye color there, right? You just have different alleles. Meiosis separate them, makes two packages, and the separation is kind of random as we learned in meiosis. So if all the copies coming from one parent don't end up in one gamete, and all the copies from the other parent end up in another gamete from an individual, it's mix and match, right? Sometimes the parent one copies from chromosome pair number one will be chosen, but then chromosome pair number 2 through 6 is parent 2, and all of those end up in the same gamete. So I hope we just clear on that in meiosis how those homologous chromosomes get separated. And when we talked about the Punnett square before, we just chose one particular gene and looked at the consequences for offspring of mixing, matching the different gametes from different parents, right? And that's only one gene, but we're not fools here. We know that during fecundation, gametes come together, and each of those gametes contain the 23 chromosomes, right? So they have all of the genes at once. So how do you do for more than one gene? Well, we start slowly. Wow. Cuckoo clock break. So we start slowly and we think about two genes. And that's what a dihybrid cross is when you look at two genes. So if you think of a dihybrid cross that provided to you in the crossing, you can, in the PowerPoint, it's slide 27, I believe, that I'm looking at, 28, sorry. When you look at that slide, you have some of a crossing that can be simplified, right? Because if your gametes are all the same, you don't need to have 4 rows and 4 columns. You can just simplify it. That's the one on the left on that slide. But now, if you cross in a dihybrid cross, dihybrid means two, right? So two genes, and hybrids means you heterozygous. So now the parents, they don't have homozygous genes. They have, in the example that is given to you, one parent is heterozygous, right? And the other parent is the same, heterozygous as well. So what happens when you cross those two? And that's why a dihybrid cross is all about. Hybrid, heterozygous, and D because you have two genes, the R gene and the Y gene. So the one for wrinkles and the one for coloration of seeds. So when you do that cross, you build that Punnett square, and I really encourage you guys to build that Punnett square at home with your bare hands, drawing on paper, and literally just starting with that, choosing the gametes that each individual can make. So if you look at one individual here, what are the different types of gametes that can be produced by one individual? That's a genotype of individual. Of course, that individual has way more than two genes, right? So we're going to go look at only two genes for now. But if we look at those two genes, well, that individual, when you separate the homozygous chromosome, it means you separate those two alleles from each other and those two alleles. So you can take the big R and put it with big Y. You could also have a big R with a little y. You could have a little r with a big Y, and you can have a little r with a little y. You cannot have a gamete with big R, little r, because meiosis separates the homologous chromosome. So you cannot have two copies in a gamete. Remember, gametes are haploid. They only carry one copy of each gene. That's the whole point. It's otherwise when you end up, if you end up with a gamete like that, that's how you end up with trisomy 21st, for example. The homologous chromosome pair 21st didn't segregate. And so you have two copies of those genes that are carried, and now you have a gamete with 3 chromosomes instead of 2 for that homologous pair. Okay, so those are the different types of gametes. Of course, that's the same individual, so same types of gametes. Those end up rows, headers and column headers in your square, Punnett square, and then you fill it in. Really important that you do it now and on your own. I'm not going to walk you through that. That's just simply putting letters and matching them, because it would get you to the habit of doing so. also start playing with the combinations. You could just say, hey, what if it's YY, big capitalized YY? What does that change for the square? Having that understanding building up is only gonna come when you do that type of work. I'm not gonna ask you to do it. You have to do it yourself. Like, I'm not gonna force you to do it. That's what I'm saying. I'm asking you to do it. That's the hybrid cross, but you can say, what if I get all homozygous dominant and then whole homozygous recessive? That's the one on the left in the slide 28. But now what if I switch things around and I just put one capitalized letter? What if I put like those two dominant and that's still heterozygous and all of those heterozygous, and see what happens? You need to do that over and over again, like that it becomes second nature. So that's for the hybrid cross. Now the law of independent assortment is because we won't think of it's not. It doesn't mean never happen. It never happens otherwise. But you want to think of an individual as a full individual. So when you think of that, that individual has way more genes than those two, right? He has a ton of them. Way more chromosomes than that homologous pair and that homologous pair, right? It has actually 23 if it's a human being. Now, what we mean by independent assortment is whatever happens when you make the gametes, it's completely whatever happens for RR, like which one goes where for which gamete, it doesn't influence how the other one is split. So in anaphase 1 and anaphase 2, when you look at those two steps in meiosis, that's when you separate anaphase 1 the homologous chromosomes, right? And anaphase 2 the sister chromatids. When you think of a cell in 3D, you think of a cell that is containing all of the chromosomes. So in there, in the nucleus, you have 23 pairs in anaphase 1. 23 pairs of replicated chromosomes. So when you separate one pair, you send, like we use blue and red copies to code them in the slides. In the slide now 29 that I'm on, looking at for the PowerPoint. We put the blue for one parent and the pink for the second parent. Those are the origin of a chromosome in fecundation for that individual that now is going through meiosis to make gametes. When the first pair sends the blue one side of the cell during anaphase, so let's draw it. So in anaphase, in metaphase 1, you will have the homologous chromosomes lining up in the middle, right? And maybe we're going to do only 3. Metaphase 1 has the spindle fibers, right? And the centrosomes here on either side. That's why it's so important that you know your meiosis and mitosis. That's my metaphase 1. In anaphase 1, they get pulled apart, right? Each homologous chromosome on either side. What I mean by independent assortment is that pair here, the blue goes left and the red goes right. That doesn't influence which one goes left or right for any of the pairs. And in 3D, a cell, and with 23 pairs for human beings, each of those pairs get completely sorted out randomly due to how they align during metaphase 1. That's completely random. That's independent assortment. When that is broken, that's how you get trisomy, right? So anaphase 1 separate those homologous chromosome, and then the separation of sister chromatids in anaphase 2, which is almost similar, but now instead of having 23 pairs of chromosomes, you have 23 chromosomes. And I'm just going to represent 3 of them. And you see that you can have all of them for one parent or all of them for another parent. It's completely random because it's independent assortment. When those sister chromatids are pulled apart here, each of them is carrying a copy of a gene, and it might be the same allele or it can be different allele. And it's not because if you're heterozygous for the 3, the dominant might be going left here and the recessive right. But in that case, that wouldn't be influencing the second one. So that first one, what happens between the dominant and recessive of the 2 recessive or 2 dominants, has no influence on the other one, how they split. And that's the independent assortment law. It's whatever happens for one pair of chromosomes during anaphase 1, during segregation of homologous chromosomes, or during anaphase 2 segregation of sister chromatids, that has no influence on the other chromosomes. Of course, it's a law, and it's not always going to be the case. Sometimes you will have mistakes, but it's not going to be viable most of the time. And when it is viable and the offspring is viable, it's usually a disease. So really, thinking about it the way I showed it to you really brings those things together, blinking mitosis, meiosis, and now the Punnett square and the Dihybrid cross and the law of independent assortment. Hopefully you understand better what's happening there. That's all for this one. See you next time.